They find bodies out in the woods all the time with missing lips and fingers.
I will make sure to add that in next year.
In Alaska, not here.
Emily. They do. I think rats and rodents have advanced senses of smell. They also have their vibrissae, their whiskers, that pick up a variety of different things that don't really match what we pick up as humans. So they're different, but they do have a strong sense of smell. Most other animals are better than us at smell. We talk about human olfaction. Despite our weak sense of smell, we can still pick up over 10,000 chemicals. Again, we're talking chemoreceptors here. And smell, again, just sort of like taste, has an evolutionary component where it has really served to protect us. So most of the things that we can smell are noxious stimuli. Not that that's what we encounter every day, but the majority of our potential smells are things that are noxious. So not only does smell contribute to our enjoyment of things like food or like being outside in the springtime, it also protects us. So we can smell smoke or gas if it's in our house. We can smell if our meat or our milk has been spoiled. So it serves a protective function in that way. And we didn't talk about it really with taste, but with practice, smell can get better. Again, probably a plasticity thing where more areas of the cortex are being devoted to perception of smell, but you have professional perfumers who train their nose to be able to distinguish and discriminate between the different parts of a smell. Just like wine tasters who both work on smelling the wine and the different flavors and the different taste, sorry, I shouldn't say flavor, the different taste within the wine. So this is an overview slide, and then we'll sort of break it up, but just to orient you, smell is going to come in through the nostrils, and it's going to make contact with this area called the nasal epithelium. And within that nasal epithelium is a layer of mucus, which is where the receptors are going to be located, and those receptors are attached to a bipolar cell, which are the yellow ones here. Those cells are going to continue their axon up through the cribriform plate, the bone in the upper part of the nose, and synapse on a second-order neuron in the olfactory bulb. So here, what you're seeing are branches or filaments of the olfactory nerve, of cranial nerve 1. Cranial nerve 1 never comes together and just travels uniformly as one nerve, like most of our other nerves do. They're in these different pockets or different gaps in the cribriform plate, and they travel up and synapse on the second-order neuron again in the olfactory bulb. So we're going to break this down, but that's the general pattern of how smell, how odors move from the periphery into the central nervous system.
Did you say that the first-order neuron is the bipolar cell?
This first-order neuron is the bipolar cell. So these yellow cells with the receptors being down here on in the mucus layer of the epithelium, sending information up all the way through to the end of the axons here in the olfactory bulb. Okay, so if we look just at the olfactory epithelium, this is the inside, the covering of your nasal cavity, which is about 25 square centimeters, covering that amount of space. Just like taste receptor cells, cells in the nasal epithelium can regenerate either on their own or following damage. But there is a natural turnover of these cells about every 4 to 8 weeks. So not as often as taste buds, but still an ongoing regeneration of the cells. And within the nasal epithelium, there are 3 types of cells. We've already talked about the yellow ones in the diagram, which are the bipolar cells. Those are the sensory neurons, that first order neuron. You You also have supporting cells, which here are labeled as the light pink. And those serve as, or act like glia. They aren't considered glia officially, but they act like glia to provide nourishment, some level of protection. They detoxify chemicals. So it's like a first order protection in the nasal passageway. So as you're breathing in that formaldehyde, these support cells are going to kill some of those toxic chemicals before they reach anything else. And those supporting cells will also help to produce mucus. So again, just like glia and other parts of the nervous system that we've seen, these support cells nourish, they protect, and they produce mucus. And the third type of cell are the basal cells, which are like stem cells. So that's how you can produce new olfactory neurons. And there was something equivalent to the basal cells within the tongue as well. We just didn't talk about them. But cells that allow for the generation of new taste cell receptors. And that's going to start here at the basal cells. So again, just looking at this, those axons from the bipolar cells are going to come up, and these groupings are the filaments of the olfactory nerve. Unlike most of the sensory systems we've seen, there is no olfactory ganglion. Okay? So there's no, the somatosensory system had the dorsal root ganglion. The trigeminal somatosensory system had the trigeminal ganglion. We just looked at ganglion related to taste. There is no ganglion for the olfactory system. They just travel up and synapse within the olfactory bulb. So we'll go up in a minute. We'll focus on the receptors, which are down here in the mucus layer right now. So when we look at smell receptors, they're located in the mucus of the nasal cavity, and airborne odorants dissolve within the mucus and bind to protein receptors on the membranes of those receptor cells. So we're not going to talk about this chemical reaction again, just like we saw before, but like we've seen pretty much in every other cell, some type of chemical reaction happens, and it's going to affect gating of either the calcium or the sodium ion channels to depolarize the membrane. So a chemical reaction leading to depolarization, just like we saw with taste. One fun fact, I guess, is that these receptor cells habituate relatively quickly to continued stimulation. So meaning that after you've been in the formaldehyde room for a little while, you stop smelling it as acutely as you would, as you did compared to when you just walked in. So our smell receptors have their own mechanisms of habituation. We can habituate in other ways. Our cortex can tell us, hey, stop paying attention to this now, you've already figured it out. So that's one way we can habituate. But the receptors themselves will stop responding after an odorant has been around in the nasal cavity for a certain period of time. The receptors themselves may respond to only a single odorant. So we see that here with number 14. It is only responding to this odorant G. Or we can have receptors that respond to a pattern of odorants. So here, this receptor responds when there is a pattern of E, G, and H, or 11. Responds when C and E are present together. So there are over 350 types of receptors. So we're able to smell or distinguish a variety of patterns of chemicals, either in isolation or when they're coupled together. So you can see how this level of organization or specificity is sort of like that other types of like somatotopic and retinotopic organization, that we have a certain level of organization in the olfactory system. We don't call it olfactory-topic or whatever, but it's that same kind of idea that we're going to have these certain receptors responding to certain patterns or certain types of chemical stimuli. And we'll see in a minute that those will organized as they travel up to the olfactory bulb. The sensory neurons, we're back to these again, are the bipolar cells. They are chemoreceptors. And the cilia, the receptors are on cilia that are in the mucus lining. So we've kind of said all of that already. Collectively, the olfactory axons are the cranial nerve 1, or the olfactory nerve, and they will travel to the where? Olfactory bulb. Okay. This is just another way of showing that the axons, the cells themselves, are only going to express one type of olfactory receptor. So the receptors are here in the mucus lining. They are going to respond to either a single chemical or a pattern of chemicals, and that signal is going to be sent through that cell on one specific axon. So it's like the labeled line theory that we saw for taste. One receptor is specific to one type of smell. That axon is going to just carry that type of smell, or that pattern of odorants. And that organization is retained when you get to the olfactory bulb through these structures called glomeruli, which are the circles here. They're labeled as number 5. And so smells of pizza, that combination of odors, are all going to synapse on the pizza glomeruli. And the flower smells like roses are going to all synapse on the rose glomeruli. So there is a high level or precision of organization that's retained at the level of the olfactory at the olfactory bulb. So let's look at the whole thing. So those axons from the olfactory nerve are going to come up and synapse within the glomeruli on these cells called the mitral cells. Right? So that's the blue, the red, and the green. These are your second order neurons, or mitral cells. Sometimes they're called tuft cells, Tuffed, but they're usually mitral cells. It's the mitral cells that are going to form your olfactory tract, and it will travel to higher level brain centers. Some of this is repetitive, but again, looking at the glomeruli, the important thing to remember is that it's a very precise level of organization. That this is a cartoon showing 4 different receptor synapse pathways. Remember, you have about 350 different types of receptors, so there's going to be glomeruli that are associated with each one of those. So highly structured, highly organized, and when a new olfactory cell is born, down here, the glomeruli will send out chemical signals which tell the axon how to grow and where to connect. Because you're going to have your cell body here, and the axon is going to have to grow. The glomeruli is what's going to signal, hey, you need to connect here. This is the right synapse for you. Okay, so olfactory pathway, let's get it done. Let's get into the brain. Your olfactory nerve fibers are going to synapse, so this is your first order neuron, are going to synapse in the olfactory bulb onto mitral and tuft cells. Okay, there's only one F, I think I spelled it wrong for you. From there, they're going to travel in the olfactory tract, and then either follow one of 2 pathways, the lateral olfactory pathway or the medial olfactory pathway. If we look at it here in the diagram, that first order neuron from the olfactory nerve filaments are going to synapse on the mitral cells, or the tuft cells, in the olfactory bulb. These are second order neurons traveling via the olfactory tract. And the blue ones here are representing the lateral olfactory tract. And that's our primary olfactory pathway. And then a smaller pathway here is represented in red. And we'll all talk about these different connections in a second. So the lateral pathway, come back, is in blue, is going to go via the olfactory tract to 3 places. entorhinal cortex. So that's what you have here on these bullets. So the first one is lateral olfactory tract. It's going to travel from the olfactory tract to the piriform cortex. The piriform cortex is located between the insula and the temporal lobe. It's going to go from the piriform cortex to the thalamus, to the area of the prefrontal cortex that is our olfactory cortex. I'll talk about that in a minute. But that's one pathway on the lateral olfactory tract. Piriform cortex to thalamus to prefrontal cortex. You can see that here. Lateral olfactory tract, piriform cortex, thalamus, prefrontal cortex. You'll see in the diagram that some fibers will bypass the thalamus and go straight to the prefrontal cortex. You want me to worry about that? Sorry. Another pathway within the lateral olfactory tract goes directly from the amygdala to the prefrontal cortex. And the third one is going from the entorhinal cortex, which is an area of the brain it's in the medial temporal lobe, sort of between, it's around the hippocampus. The structure associated with memory, just like the hippocampus. So it's going to go from the entorhinal cortex to the prefrontal cortex, then to the hippocampus. So you'll see the structures here. Unlike any of the other sensory systems we've seen, the olfactory system does not synapse in the thalamus before going to the cortex or to other regions of the brain. So unlike any other sensory system we've looked at, the olfactory cortex is the only one that doesn't make a pit stop at the thalamus before going on to higher level brain centers. It's going to go first to areas of the brain that are associated with learning and memory and emotion. Yeah, Katie.
That's a question. So when you like smell something, it goes to the olfactory bulb and then down the tract.
Correct.
So the olfactory bulb, like on our gross brains, it's just like loose. So how is it connected? Does it just come unconnected?
It's going to sit in a fossa on the skull, so when you put that brain into a skull, it's going to have a resting place within the skull.
Okay, and so the synapses, the nerves are going
Up,
straight.
up through, oops, I'm going the wrong way. Up through
Okay, so that's
this bony layer.
okay.
Yep,
Thank you.
good. Okay, so those are your three lateral pathways. Those are all, oh, sorry, go ahead.
So with the lateral pathways, so would the third order neuron be in the piriform cortex, the amygdala, and the
Yeah, the cell body of the third order neuron would be in one of these three structures.
Okay, thank you.
Jamie.
So you're saying it doesn't make a pit stop with thalamus, or just doesn't synapse with thalamus?
It doesn't go there first.
Oh, it goes to the cortex.
So everything else we've seen kind of comes in at the brainstem or at the spinal cord level and goes and hits the thalamus before going to the visual cortex or the somatosensory cortex or whatever. The olfactory system will stop first in other parts of the cortex that aren't higher level brain centers, that are limbic structures or whatever. It's not going first to the thalamus. It will, in this case, go to the thalamus, but not, it's going to stop here first. Yeah, Paige.
When's the synapse from the second to the third order neuron?
It's going to be in one of these three structures. Okay. All of this lateral olfactory tract is ipsilateral, but some fibers will cross over via the anterior commissure. And I know you've been waiting since week 1 to know what the anterior commissure does. So this is one thing. is it carries olfactory sensations from one side of the brain to the other. Okay, so in the diagrams, in the slides, I said all of this is going to end up going to the prefrontal cortex. It goes to a specific area of the prefrontal cortex called the orbitofrontal cortex. And here's a mid-sagittal view, and here's an inferior view of this part of the brain. And this is a big area for integration. So it's not just the olfactory cortex. It is the place at the level of the cortex where smells are going to be identified, where we start labeling, like, oh, that smells like a rose, or that smells like a tulip. It's where we're going to start distinguishing or having higher levels of discriminatory abilities about smell. But some of that's going to happen at lower level centers too. But all of the smell information is going to come here, and we're going to identify it, and we're also going to associate a reward value with it. So sort of a hedonic pleasure associated with that smell, or an evaluation of what this means to me, what this means in the greater sense. What is this worth to me? And that'll make sense when we talk a little bit about decision making. Similarly, this part of the orbitofrontal cortex is a secondary processing center for taste. And it's not our primary gustatory cortex. What were the two parts that made up our primary gustatory cortex? Insula and frontal operculum. Right, don't get it confused with pars opercularis. It's the frontal operculum. It's that underneath part. So this part, the taste processing that happens here, is again associated with reward value. So information about, again, how much is this worth to me? How much do I like it? And we'll talk about, again, give me one more thing before I start talking about that valuation. The other type of sensory information that makes it here is information from cortical visual areas. And really, it's that stream, that ventral stream that goes through the temporal lobes that comes here. And so that's telling us about what? The what, right. What is that object, right? Ventral stream is the what. So in this area of the brain, we start linking what you smell and what you taste to what that thing is, what it looks like. And you start saying, what is that worth to me? You know, do I want that extra piece of pizza? Is it worth it to, you know, have that extra beer or that glass of wine? We start associating, connecting things. So it's an area of sensory integration, but it's also an area where we have stimulus association learning, where we associate a stimulus with another stimulus, and then we add a level of value to it. And that level of value helps us to make decisions based on expected reward or reinforcement. So if I have that extra beer, am I going to feel really great? And I'm going to be feeling, really good for the rest of the night? Or if I have that extra beer and I taste that beer and everything, am I just going to feel lousy? Is it going to tip me over that point of feeling good? So again, prefrontal cortex, we talked about that with teens. It's big in making decisions. And taste and smell are two of the sensations that really contribute to that area of the brain. So if you guys are studying for your neuro exams, like how many of you guys will use food as a reinforcement? Like if I get through this stack of note cards, or beer, I can go down and have a beer, or I can have that extra piece of pizza. So that type of training yourself and that type of sort of patterning of behavior is going to start in this area of the cortex, this area of the prefrontal cortex. And unsurprisingly, this area is also heavily implicated in addiction. So you're adding value to a chemical or a type of drug interaction. It's not the only place that's responsible, but it is implicated in addictive behaviors. I believe it's also implicated, and you don't have to know this for anything, but a fun fact, I think it's an area of the brain that's really been heavily identified suicide, and obsessive compulsive disorders as well. So yeah, interesting. Somebody, Meg, was that you?
I was just gonna say, is this the same area that would have like emotional eating tendencies as well?
Yes, I think so. Yeah, it's and again, there are other areas doing that as well, like your amygdala. But yeah, I mean, everything kind of comes together here, and so your amygdala is going to associate the emotion with it, but this part of your brain is going to say, do I do it or not?
Is that, like, we talked about like strengthening synapses and stuff, so if you do that more, do you strengthen that pathway or
If it works out the way you predict, so if you drink that extra beer and you feel really good afterwards, you've strengthened that pattern of learning. You're more likely to do it again. But if you anticipate something's going to happen and it doesn't happen that way and you end up barfing in the street, maybe the next time you don't do it. You've associated less value to that extra beer, right? Maybe. There's a lot of ways you can justify drinking for neuroscience, right? Because we talked about last week with dizziness and the vestibular system. Now we've got it with reward and reinforcement. Okay, remember there were two different pathways. That smaller medial pathway is here in red. That, again, is going to bypass the thalamus, and it is going to travel via the anterior olfactory nucleus. So where is that? Anterior olfactory nucleus, contralaterally. So this one is going to cross over and go to limbic structures in the basal forebrain. So the basal forebrain isn't an area we've talked about. It's an area that is around the caudate and the putamen, so around the basal ganglia. So the basal forebrain is an area that is around, in front of the caudate and the putamen. And again, it is as a set of structures. Those structures are associated with learning and memory. So this is another path, another example of how smell ties in closely with our ability to learn and remember. And whether or not we use these pathways in the same way as they were evolutionarily designed, like most of you guys don't think about sitting down to study and smelling your notes, right? So we're not using our smell a lot for learning and memory, but there is that association. We tend to associate our grandma's house with a certain smell, or our homes, our bedrooms with a certain smell. Even the pheromones that you're attracted to, or that people release, we have associations with smell. So there is that concept of learning and memory and smell. We probably don't have to use that as much for survival as we used to, because we used to have to use smell to, again, locate food sources, determine if the food source was viable. I don't think these are as important as they used to be in our evolutionary history. But we're not that old as a species. So maybe some of these things will start to go away as we evolve, and other things will take their place. Do you have a question? All right. Almost done. Again, as a helpful hint to you in pronunciation, these are osmias. Oz, get it? Okay. So, lack of smell are osmias. So you have anosmia, hypoosmia, I don't even know how to say this one, cacosmia, that's it. Dysosmia. Okay, so that is your land of osmias, all referring to either a loss of or a distortion of smell. So anosmias or hypoosmias can be temporary or permanent, and they can be caused by just things like the common cold or flu, some sort of irritation to your nasal lining. You've got sort of swelling in that mucosal layer. The receptors aren't going to be as sensitive. They're going to be blocked. So that is sort of the osmia that you are most familiar with, which is hypoosmia, or decreased ability to detect odors. Again, those are temporary. They can also be caused by something that's physically blocking the airflow. So like a nasal polyp or a tumor that is blocking those areas, you can have a disruption, again, either a complete loss of smell, more likely a decreased ability to smell. So again, tumors, nasal polyps, any type of bony deformity within the nose can cause a loss of smell. Cacosmia are hallucinations of unpleasant smells, usually associated with seizures, sometimes associated with brain injury. So people with cachosmias experience like really horrible smells in the absence of anything else being there. So often those smells are like rotten egg smells, odors of like rotting fish or garbage. They're just really nasty smells, and they're considered again hallucinations because there's nothing that's actually there. So they're not misinterpreting smells. That would sort of fall under altered perception of a smell that normally wouldn't be unpleasant. But a true olfactory hallucination, and they're usually very bad smells. So you're not smelling roses in the absence of anything else. It's really a bad smell. Again, most of the time this is a result of an injury to the olfactory pathway, either due to seizures, your book talks about uncinate fits, or brain injury of some sort. You don't really have to worry about all these. I put them on there for your future reference. If you are working with people that have distortions in their sense of smell, you can have phantom smells and agnosia, the ability to smell but the inability to classify it. So if you run into that, you have this in your notes for future reference. Tara?
Just for clarification for the C ones, associated with seizures, so they have seizures and now they randomly have these hallucinations, or they get these smells before they've had a seizure?
In this case, they're related to damage that has occurred because of a seizure. But I do think I have heard of people who have seizure disorders get a phantom smell before or have a sense that they're going to have a seizure. And I believe that's different. You might want to check in your books about that. But I think in this case, they're talking about damage that is due to either seizure or brain injury. Okay. Other things that can affect olfaction, any type of physical damage like a head trauma, again, seizure disorders, Parkinson's disease, and some of the others like multiple sclerosis or Alzheimer's, because you're actually losing neurons or losing the ability to transmit signals, different types of endocrine disorders. We aren't going to go through the whys of all of these. Again, this is more of a FYI for your future clinical practice, that if you are working with people that have any of these conditions, smell may be part of your initial battery of testing, or particularly if you suspect sensory loss, then you could try and evaluate their sense of smell. There are standardized tests to do that. What I will say is the way that this is going to affect occupation, I'll ask it. How? Why do we care if they can smell or not? Safety. Number one, safety. So if you're sending them home alone and there's a fire in their kitchen, are they going to be able to smell the smoke? Or if they can't smell the smoke, is something else in place, fire alarms or visual reminders or whatever, that are going to alert them to that danger? Are they going to go home and eat rotten food because they can't smell the difference between good milk and bad milk, or good meat and bad meat? So your role is really just to assess the safety of that person related to their sense of smell. And with taste and smell both, are loss of taste and smell going to affect nutritional value? So we talked about picky eating. We think about that mainly with kids, but in older adults as well. Are they eating enough? Are they getting enough calories? Are those calories nutritious enough that they're getting the vitamins, the minerals that they need to be healthy? Okay, that's it. Taste and smell. Questions on that? I will say I'm not going to ask a lot of questions on this on the midterm, on the lecture midterm. So I would say, and I'll send out details on how many questions and all of that. I would expect one or two, but I'm not going to expect you guys to just absorb all of this new material a week before. Well, you actually have 2 weeks, but it'll be minimal compared to the sensations that we spent more time on, like somatosensory, vision, and auditory vestibular. Okay, so that's taste and smell. Here's your review of cranial nerves. See how many you can write down. Name them. So you're just naming them, not giving their function. couple slides, but it's olfactory, optic, oculomotor, trochlear, trigeminal, abducens, facial, vestibulotrochlear, glossopharyngeal, vagus, spinal accessory or accessory, and hypoglossal. And out of all 12 of those, really it's only the last two that we haven't talked about at all. Because even the motor neurons for the eye, the trochlear, the abducens, and the oculomotor, we touched on. So there's really only two that are new at this point. Here's an acronym. I think you guys already had this. If you need something to help you remember their names. Now identify if those neurons are sensory, motor, or both.